Today, in video 9 of our DNA unit, we're going to talk about our last topic, which is gene expression. So basically, this video is going to be all about how cells know when they need to transcribe and translate a certain gene. In other words, how do they know when it's appropriate to express or create a certain protein? Because all cells don't need all of the proteins all of the time. So, thus far, when we've been talking about a gene, we've been keeping the idea of a gene pretty simple. But in reality, genes are more complex than what we've already seen. So here is kind of the typical structure of your average gene in a chromosome. And you can see this section right here. This is the section that I actually transcribe or copy and make into RNA, and then use that section to make protein in translation. But there are other pieces to a gene, and these other pieces are what we call regulatory sites. These sites regulate or decide when this gene is going to be copied into RNA, when we're actually going to transcribe it. So you can see there's these regulatory sites. There's another regulatory site right here. This happens to be the regulatory site where RNA polymerase is going to bind when it starts transcription. So kind of the takeaway is that there are other pieces of a gene than what we've talked about thus far. Okay, so what is gene expression? So we know that we have over 100 trillion cells in our body, and each of these cells is going to need different proteins at different times. So in other words, it would be a huge waste of energy to make every protein in every cell all the time. So things like a brain cell is going to need completely different proteins than a nerve cell does. So even though these two cells have the same DNA, they have the same instructions, but they need to use different parts of the instructions to make different proteins. So gene expression, we're going to define as the activation of a gene to transcribe it and make RNA, and thus make protein. So it's important that we regulate this. We want to turn transcription on and off of certain genes at different times. If gene expression goes wrong, if I have too much of a protein or too little of a protein, we can run into huge problems. Think back to our cell unit when we talked about, remember those proto-oncogenes? If they got stuck on, so we were always creating those cell cycle promoters, remember we got too much growth and that would lead to cancer? Or remember if we got too little expression, if I never turned on those tumor suppressor genes that were supposed to suppress growth, again, we can get cancer. So gene expression and this regulation of when things get transcribed is a very important process, and also very complicated. So we are going to only look at one example of one gene. We're going to look at expression of basically the protein that breaks down lactose in E. coli. So I want you to have an appreciation that every single gene is regulated differently, and we're only looking at one example. So that will give you an appreciation of how complicated this system actually is. So the two scientists who studied this example, Jacob and Menard, in 1965, basically they were looking at a protein in E. coli, a bacteria, and this protein breaks down lactose. So it's the protein that if the bacteria eats lactose, it breaks it down into glucose and galactose and uses it for energy. So this enzyme that breaks down lactose is called beta-gal, which is short for beta-galactosidase. So remember, it's breaking down lactose. The enzyme is going to break down lactose in order to produce glucose and galactose, and that glucose can then be used in cell respiration in the bacterial cell. So this enzyme is doing a hydrolysis reaction. It's breaking that disaccharide into the monosaccharides. So you can kind of think that the cell would not want to have this enzyme active all of the time. Because if there was already plenty of glucose available in the environment, there would be no reason to waste energy breaking down lactose, because it could just use the glucose that was present. Similarly, if there was no lactose around to break down, it would be really stupid for the cell to waste its energy making this enzyme. If there's no lactose to break down, you don't need it. So therefore, E. coli has to have a way to turn on and off creation of this protein. It has the gene to make this protein, and it needs to turn on and off the ability to transcribe it and then translate it into protein. So this is what the lac operon is. So first let's define an operon. So an operon is a group of genes that operate together. That's where the word operon comes from. They operate together. So an operon includes all the regulatory elements, so all the pieces that regulate the gene and the gene itself. So if we look at this operon, we can see there are 3 genes here, and these are the 3 genes that actually code for the mRNA and the protein that makes this enzyme. So these are the 3 pieces that actually will make the mRNA that will code for this particular protein. And then I have a bunch of regulatory sequences. So of all these, I want to point out 2 to you. I want to point out the promoter. We already know, remember, the promoter is where RNA polymerase is going to bind if it's going to transcribe this gene. So this is where RNA polymerase will bind it at the beginning of transcription. And then the other one I want to point out is this operator, because that's going to become important later. Okay. So this just breaks down what we just kind of talked about in that picture. So remember, the first piece we looked at were the structural genes. So this was the actual part of the gene that codes for the enzyme. It codes for the beta-galactosidase that's going to break down lactose. We had that promoter, the section of DNA where RNA polymerase binds to start transcription. So that was a regulatory element. And then we also had that operator that I pointed out. And this is a piece of DNA that's going to act like a switch. This is the section of DNA that determines, yes, transcription happens now, or no, it doesn't happen now. This is going to be the regulatory element. It's going to regulate if we transcribe or we don't transcribe. So now we're going to look at the situation. We're going to look at a situation where we want to transcribe and one where we don't, and we'll see how the system operates differently. So here, first we're going to look at a situation where there's no lactose. So kind of think, if there's no lactose, I do not need an enzyme to break down lactose, so I would not want to transcribe and translate this gene. I would not want to make this protein if there was no lactose. So the way this works, the way the system for just this gene works, is there's a regulatory gene right upstream of the gene we're looking at, and upstream just means before. So before, in an earlier section of DNA, there's a regulatory gene that will create this protein. So it will make a little mRNA, and it will make this protein called the repressor. And the job of the repressor is to bind to that operator, or bind to that switch section of the DNA. And the idea is, if the repressor is bound to the operator, so you can see my protein is bound to the DNA here, RNA polymerase comes along, and RNA polymerase wants to transcribe the gene and make my protein. But if the operator is bound, if the repressor is bound to the operator, my red protein here is literally in the way. So RNA polymerase is not able to get to the promoter. It can't get there, so it can't bind. So therefore I get no transcription, because RNA polymerase can't bind to the promoter, can't bind to the DNA. So if the operon is off, and it would be off because no lactose is present, that means I do not want to have transcription. Transcription is turned off by this repressor protein. So the repressor binds that operator, that regulatory section of the DNA, and it physically prevents the RNA polymerase from binding to the promoter. So therefore, I do not create my enzyme. I don't create my mRNA and don't create my protein. So now let's look at the opposite situation. Let's look at the situation where I do want transcription. So now, if lactose were present, well, now I do need the beta-galactosidase. I do need that enzyme to break the lactose down so I could use it. So the same thing is going to happen. My regulatory element upstream of my gene is going to create my repressor protein. But now, this is where it gets a little different. So remember, lactose is present. So look at, lactose is hanging around here, and lactose can actually bind to the repressor protein. So when it binds, it changes the repressor protein's shape. So now the repressor protein can no longer bind to the operator. So I'm putting a big X here. This protein's shape was changed. It cannot bind to the operator. And because it can't bind to the operator, well, now RNA polymerase can bind to the promoter. It's not in the way anymore. And if RNA polymerase binds to the promoter, it's going to go ahead and transcribe my gene that makes my enzyme. So when lactose is present, the lactose represses the repressor. So now RNA polymerase can transcribe. So we're kind of getting double repression going on here. So in this situation, if I want my operon to be on, lactose is present. So I need to create the enzyme that will break down lactose. Lactose itself, it's already present, we just said that. It's going to repress the repressor. It binds to the repressor, changing its shape, and making it so the repressor can no longer bind to the operator. And if it's not bound to the operator, RNA polymerase is free to bind to the promoter and transcribe the genes. So we would say the lac operon here would be on. I would be making my beta-galactosidase protein. So you could see this system is super complex, and I'm telling you, there's actually other pieces in this very system that we're not talking about. So gene expression is so complicated, and every single gene in our body is regulated a different way. So we kind of really gain this awesome appreciation of how cool the body is and how it's able to really decide when we need proteins and when we don't. So, I mean, this is one of the things that I love in biology. It's so neat. So we can see that the situation is very complex, and that we're kind of very lucky that it all works and our bodies work the way they do. So that's the end of the DNA unit. See you in the next unit.